Hi everyone, my name is Hamid Asadi and I'm a fourth-year PhD student working in non-electrochemistry lab in the College of Engineering at the University of Georgia. Today I'm going to talk about electrochemical biosensor for detection of microRNAs as new cancer biomarkers. Despite recent technological advancements, the rate of survival in cancer patients is still low, which is mainly because of diagnosis of cancer at very late stages. Identification of certain cancer biomarkers can help us in early diagnosis and subsequent treatment of cancer. These biomarkers can be found in most of body fluids, which makes them attractive alternative to invasive biopsy methods. Here, we focus on the detection of microRNAs as newly emerged cancer biomarkers. There are many scientific evidences that the dysregulated expression of microRNA compared to the basal level in body fluids can be closely related with the occurrence or diagnosis of cancer. Conventional methods of microRNA detection suffer from many intrinsic limitations, such as the need for complex procedures and expensive instrumentation. In contrast, Biosensor can overcome all these shortcomings and offer significant prospect for point-of-care testing. In this work, we present a novel label-free electrochemical biosensor for sensitive and selective detection of microRNAs as potential cancer biomarkers. MicroRNA 21 was selected as a model analyte due to its overexpression in the earliest stage of prostate cancer. To fabricate the biosensor, a glassy carbon electrode was first modified with a thin layer of graphene nanosheet. Then, the captured DNA probes were covalently immobilized onto the electrode surface using a molecular linker called PBSE. The electrochemical detection of target microRNA 21 was achieved through measuring the changes in the interfacial impedance of the electrode surface following the hybridization events between microRNA 21 and the pre-immobilized complementary DNA capture probes. Different variables related to biosensor fabrication and experimental conditions were optimized to acquire the highest biosensor response and the shortest detection time. As can be seen, a linear relationship was observed between the charge transfer resistance values versus the logarithmic concentrations of microRNA 21 within a concentration range from 10 to the power minus 14 to 10 to the power minus 8 molar. From that, a detection limit of 3 femtomolar was calculated for the biosensor, which is low enough to detect microRNA 21 present in human body plasma and serum sample. In terms of selectivity, the impedimetric response of the biosensor toward non-target microRNA 141 was about 80% lower than that obtained for the complementary microRNA 21 at the same concentration, which confirms an accepted level of selectivity. We also evaluated the storage stability and the applicability of the biosensor for detection of microRNA in real plasma samples. The biosensor was able to maintain more than 90% of its initial ability for detection of microRNA 21 for a wide range of concentration, even after being stored for 14 days in immobilization buffer. Besides, the obtained recovery values by the biosensor in diluted plasma samples varied from 90 to 116%, which were all within the clinically acceptable recovery ranges of 80 to 120%, and clearly showed that the proposed biosensor has a great potential to be used for sensitive and reliable microRNA detection in clinical diagnostics. Altogether, the results of this study indicated that the proposed biosensor could be used as a promising alternative to conventional methods in early screening and clinical diagnosis of cancer in a point-of-care setting. Thank you so much for your attention.